So in this quick example question, we're wanting to look at basically how we can apply our knowledge of Western blotting to come up with conclusions for this Western blot. So we're told that we are looking at protein kinase C alpha, and protein kinase C alpha has a molecular weight of 80 kilodaltons, and that can be seen in the Western blot here. So the question is telling us that, well, it's asking us to make a conclusion, and we can see that Western blot analysis of extracts of HeLa, COS, C6, and NIH3T3 cells using an anti-PKC rabbit antibody are used to make this, to yield this plot here. So what the heck does this mean? Well, this is just these are our 4 cell lines. These are just different cell lines. It really doesn't matter actually, because this question doesn't ask us anything in particular about these, specifics. Like, it doesn't need any prior background knowledge. All we're doing is looking at this and coming up with a conclusion. So what we can see straight away is that, I should probably mention, this is our molecular weight ladder. So this is used in all Western blots, and it allows us to make an approximation visually after we run our blot, because we can see, for example, with this question here, that there are, so we know that protein kinase C has a molecular weight of 80 kilodaltons, and so for that then we can look at the 4 different cell lines, and we can see 4 bands that are all at 80 kilodaltons. So this would mean that we have protein kinase C alpha in all 4 of our cell lines. Now obviously some of them are a bit larger and some of them are a bit weaker signals, so that's what we are going to discuss. So we can see basically the HeLa and the C6 cells have relatively the same signals, which means they have probably the same amount of PKC alpha in their cells, whereas COS has the weakest signal out of the 4 cell lines. So that would mean, well, what does this mean? Remember that this is picking up, our Western blot is picking up basically how much protein was in the cell. So whilst yes, there was the least amount of protein kinase C alpha in the COS cell, that's because, well, not because, but we see that the signal is weakest because there are fewer antibodies still present in this cell line. Why would there be fewer antibodies? Well, we know that the primary antibody, which we can see is made from a rabbit, this is obviously going to bind to our PKC, and then when we add our secondary antibody, which would probably come from a goat or a donkey, that secondary antibody is complementary and specific, and therefore will bind to a primary rabbit antibody. So if we have less of the protein of interest, our protein kinase C alpha, it means that we have less primary antibodies binding. So that means when we rinse the membrane, we actually lose these primary antibodies. And so when we add our secondary antibodies and then we wash them, well, we're just going to lose the majority of these secondary antibodies, because there's not very much primary antibody to bind for the secondary antibodies to bind to. So that's why actually COS has the lowest signal, because there is less protein in it, basically. But you need to be able to understand in terms of the primary and secondary antibodies. And hopefully you recognize that the signals that we receive during Western blotting are coming from, well, 3 different types of things. It can either be a chemiluminescent result, or maybe colorimetric, or perhaps it was fluorescence. So in the case of, let's say, a chemiluminescent Western blot result, that would come from the secondary antibody being conjugated with something like horseradish peroxidase, which actually catalyzes the reaction of hydrogen peroxide with luminol. And so the reaction of this is activated luminol, which gives, well, is luminescent. And it's that light that is picked up by a digitizer or a photo film. And that's how we get these results. And finally, we can conclude that this cell line here, the NIH3T3 cell line, has the greatest amount of protein kinase C alpha within it, and that's represented by the large mark basically at 80 kilodaltons. So I have actually just gone into detail on how we've gotten these proteins, how we've gotten these antibodies, but this is just telling us a bit of background information. So it says this antibody is from CST, and the primary polyclonal antibodies were produced by immunizing rabbits with a synthetic peptide corresponding to human protein kinase C alpha. So what does that mean? I did say this before, it was a bit convoluted, so I'll quickly just say it again. What do we mean by immunizing rabbits? So that's when we are taking a specific amino acid sequence from our target protein. Again, in this case, it's our protein kinase C. And we're really taking this sequence of amino acids from a region called an epitope. And then what we can do is we can inject this sequence of amino acids, this peptide sequence, into a rabbit. Then the rabbit recognizes this as non-self, so it will produce antibodies that are specific to the peptide sequence that we've just injected into them. Then we can take the antibodies that were produced from the rabbit, and we can then inject those into our goat or our donkey, and they will produce antibodies that are complementary and specific to the rabbit's primary antibodies that we made. Then we, like I said, conjugate that with something, with a substrate that can be used to produce a Western blot.